John very eloquently covered the promise of genomics in cancer and genetic diseases, and my role is to focus on complex diseases. And by that I mean things like cardiovascular disease, type 2 diabetes, obesity, that sort of thing. And I think Chris Levi asked me to do this talk. From a clinician's point of view, I have been a general physician for the last 20 years, but I happened to do my PhD in molecular genetics back in the late 80s and early 90s when the Human Genome Project was starting. And so I've been fortunate enough to see that through and see what it's delivered. So here goes 30 years of research experience distilled into 10 minutes. We'll see if I'm up to the task or not. So the promise of the Human Genome Project, it was one of the biggest feats of human collaboration. Started in 1990, technically finished up in 2003, but we're still getting a lot of data. Among the many things that it revealed to us was that our genome is made up of 3 billion base pairs, and there's nothing that talks about sort of our common humanity as much as seeing that 99.9% of our DNA is common between any two random people on the planet. That 1.1% difference usually takes the form of a single base pair change, what's called a single nucleotide polymorphism, or SNP for short. So 0.1% basically accounts for all the difference that we see between each of us. 0.1% spread over 3 billion base pairs, though, still adds up to 30 million differences, 30 million genetic variants. And the next bit of technology that came along was these microarrays that basically meant that on something the size of a microscope slide, you could get a handle on these 30 million genetic variants for under $200 now. John spoke about the next step, which is to do the entire sequence, but this was where we are pretty much up to so far. And it's the development of these microarrays that led to these genome-wide association studies. And so in the same way that we were used to looking at disease risk, looking at cholesterol and disease risk, blood pressure and disease risk, smoking, here we started to look at genetic variants and disease risk. And the promise in this field was that we could accurately predict disease risk. And hopefully by getting somebody's genetic profile, we could tell them what they would be at risk of. And that was based on the hope that these complex diseases would have ideally maybe 5 genes, perhaps up to 20, 30, 40 genes that could be easily managed. And if you could just profile those, you could tell people what their risk was. And the assumption was that that small and manageable handful of genes would have a strong enough effect size that they could be used clinically. I think the reality over the last 10 years has been most complex diseases turn out to be due to thousands of genetic variants, each having a very tiny effect sizes sort of 1.02 to 1.2. So the consequence of this is, yes, we can get a handle on these disease genes. They're turning out to have a lot more genes involved in each disease and a lot smaller effect size, but can they still deliver on the promise of individualized disease risk? And so let's have a look. This is a pretty typical example for type 2 diabetes. If we build a clinical prediction rule to predict who's likely to develop type 2 diabetes, using just 3 risk factors, age, sex, and BMI, we get an accuracy of our prediction model of about 0.79. We measure the accuracy of our prediction rule using the area under the curve. Here an ROC curve. We don't have a lot of time to explain the ins and outs of that, but basically you can think of it as a measure of accuracy. So 79% accuracy. If we add the top 10 genetic variants, the top 10 hits for type 2 diabetes to that clinical decision rule, basically our accuracy goes from 79% to 80%, which is really pretty unimpressive. And this is reasonably common among complex disease. Now, I stress that cancer and genetic diseases, Mendelian diseases, are a completely different matter. But in things like asthma and obesity and type 2 diabetes, it's been reasonably disappointing. Now, we just said that these microarrays give us thousands of genetic variants. So you might be thinking, why just add the 10 top genes? Why not add thousands of genes? Well, here's an example from prostate cancer. where if you build a clinical prediction rule with the top 10 genetic variants implicated in prostate cancer, your accuracy is 56%. Basically, no better than flipping a coin, not much better than flipping a coin. If we keep adding genetic information, and you see here, you keep adding up to 60,000 genetic variants, the performance of that prediction model doesn't really budge. And I think what some of this data is telling us is that we're reaching a theoretical threshold. Yes, with each gene that we're adding genetic information, but we're also adding some noise. And we're reaching a threshold where the noise is actually canceling out the information that we're adding. So if disease risk isn't going to be in the cards in the near future, where do I think, and this is just a personal view, where are the payoffs for clinical medicine in the next few years? And I just want to focus on 3 areas where the payoff might be in the near future. I think one of the biggest payoffs is in identifying new drug targets. So just as an example, there's a protein called PCSK9, which has a role in degrading the LDL receptor. And it was first identified through genetic studies back in 2003. And what they found was 1 in 40 African Americans has a genetic variant that wipes out the function of this protein. And those people had very low LDL levels. The idea being that if this protein degrades the LDL receptor, if you stop it from working, you have more of the LDL receptor, which is your bad cholesterol, expressed, and so you're sucking out the bad cholesterol from your bloodstream more easily. When we started to do the genome-wide association studies with LDL and coronary artery disease, this PCSK9 gene was one of the top hits, and that came through in 2009. And so in that short amount of time, from 2009 to 2015, pharmaceutical companies seized on this, developed monoclonal antibodies, tested them, did large-scale randomized controlled trials, and reported them in the New England Journal of Medicine. And as you can see here, 2 studies, both published at the same time, showing a big reduction in LDL and a subsequent reduction in cardiovascular disease. So I think this is tremendously encouraging that from 2009 to 2015, basically in 6 years, we've gone from a genetic signal to developing a drug to testing and publishing 2 large-scale RCTs. So that's incredibly encouraging, and I think gives us a sign of where other payoffs are likely to be in the future. I think the second payoff is, as John said, in the area of pharmacogenomics, and this is an example perhaps of how we can avoid drug side effects. So most of you will know statins is one of the most common drugs prescribed in Australia, and one of the side effects is myopathy, basically muscle breakdown. And this study in the New England Journal of Medicine was published in 2008. A very modest number of patients, 85 patients and 90 controls. These were people who developed myopathy on statins and those who didn't. And basically they identified 1 genetic variant that had a very strong effect. 1 copy of this genetic variant was enough to increase your risk of developing myopathy 4.5-fold. And if you had 2 copies of the risk gene, you had basically a 20-fold increase in the risk of developing myopathy. Now, you would think that a 20-fold increased risk of getting a side effect is a strong enough effect that we should be able to use that information clinically. But if you look at the graph, what it's showing on the x-axis is 6 years of statin use, and the top line there is the CC group, which is the group that has 2 copies of that genetic variant. And so if you look at the absolute risk scale, it's a bit small, but the absolute risk of developing myopathy, even if you have 2 copies of this high-risk gene, is still only 20%. So 80% of people in the high-risk group can still take statins up to 8 years and still not have myopathy. So I think we're going to have to be quite clever. Even when we have powerful information like this, we're going to have to be quite clever in how we use it clinically. And I think the third area is one that's been less explored, but more on the public health arena. How can we use this to prompt behavior change? So one of the tenets of personalized medicine has been, if we just give people their genetic risk profile, they will naturally change their behavior. And for those of us in public health who've been involved in smoking cessation, alcohol reduction, we know that it takes a lot more than just information to get people to change their behavior. But one of the things that people have started to think about is there is a certain cachet, a certain je ne sais quoi about your DNA. And if we can tell people, we've read your DNA, we've read your book of life, and your genetic variants tell us that you are particularly at risk of this or that cancer if you smoke or if you drink, potentially that is powerful information to prompt behavior change. And people are starting to look at this idea. How does giving people genetic information and the shape in which you give it to them, can that motivate behavior change? One of the most studied areas in this field is smoking cessation. And even up till now, there's only 6 studies so far, as you can see. And we have a summary effect size of 0.92. So it's trending towards an improvement in smoking cessation when you give people genetic information, but obviously not significant yet, but only 1,200, 1,400 people in each arm. There's still a long way to go to testing this theory and figuring out how people can understand genetic information. So I hope you'll take this with a grain of salt. This has been a bit of crystal ball gazing, so to speak. I'm not sure that gene-based disease risk prediction will come through for us. Genetic data alone, I don't think will come through for us in terms of personalized disease risk. But I think there's some other areas where we might get some payoff clinically in the next few years.